Hey there everyone. In today's class we're going to look at DNA fingerprinting. This is the last of the biotechnology series for genetics, and I thought that it would be a good idea to at least go through a little bit about DNA fingerprinting in terms of what it is, how it's made, how do we analyze a DNA fingerprint. So let's start by looking at what is a DNA fingerprint. So just like a regular human fingerprint, fingerprints are very unique, right? So a DNA fingerprint is just a unique pattern of DNA that is unique from person to person. So no one in the world would have exactly the same pattern. They may have similar patterns, okay, especially if they're related to you, but every person has a unique fingerprint pattern that's all their own, and it's made from cutting up their junk DNA. Once that junk DNA has been cut up, it's sorted into different fragment sizes. And from that, we build a DNA fingerprint. So over on the right here, you'll see that there's a DNA fingerprint, a really simplified version of it, okay, that's found at the crime scene. So someone left some DNA, and DNA forensics would create a DNA fingerprint from that DNA found at the crime scene, and they would compare it to the DNA from three suspects in this case. And when you look at the pattern, if there is a match, so when you look at where these bands are, okay, if they are identical in pattern to what you find at the crime scene, then you can say that suspect number 2 was most likely at the crime scene, whereas suspect 1 and 3 were not. So in order to figure out how the DNA fingerprint is made, okay, you do need a DNA sample. So once you have that DNA sample, it has to be processed. Okay, and the first thing that forensic scientists will do is that they'll take a very special type of enzyme called a restriction enzyme. Restriction enzymes are like a pair of scissors. They're very accurate at cutting base pairs at very, very specific DNA base sequences. These cuts are made deliberately so that long strands of DNA can be cut into shorter strands of DNA. Once you have all these different size fragments of DNA that have been cut, we need to amplify them. Okay, because typically at a crime scene, there might be a small amount of DNA that has been found, and maybe not enough to produce multiple copies of a fingerprint. So what they do is that they use the device, okay, that's pictured here, and they amplify it, okay, using a technique called PCR technique. So you just make many, many, many copies of the DNA samples that you already have so that you can continue to make as many fingerprints as necessary. Once you have all these different fragments of DNA that's been amplified, they're now put into a special gel called gel electrophoresis. It's kind of like Jello, but a little bit thicker. Okay. The idea is if you run an electric current through this gel, okay, all those fragments of DNA will sort themselves according to size. Okay. And this usually works because the DNA molecule itself is negative. So if you put an electric charge through it, okay, the ends that is negative, okay, would be pushed away from the negative terminal, okay, of this battery or this power source, and basically the fragments themselves will travel through the gel. Okay, so they literally move within the gel, and then they end up depositing at a certain point from the wells, from where you injected them. By doing this, you separate all your fragments according to size and mass, so that the heavier ones are likely to be closer to the wells, and the lighter ones, because they're so light, they can travel further away from the wells. So you'll see this in this diagram here. If we take a piece of, like this container here contains the gel electrophoresis, okay, and in the wells, you just inject samples of your DNA fragments. When you pass the current through, okay, the current will go through from the negative end all the way to the positive end. So the current is going in this direction, going towards you. The current picks up those molecules of DNA. And the heavy ones, because they're massive and heavy, they don't get to travel as far as some of the lighter ones. Okay, so in the end, you see little deposits of fragments. So each line isn't necessarily one fragment, but each line could represent several groupings of fragments of the same size. So that we see heavy fragments closer to the wells, lighter, shorter fragments further away. So if we take an actual DNA fingerprint that's being processed, at the bottom here, okay, these are the wells. So this is where you would inject your sample of DNA. Okay. Now, you see a whole bunch of thick bands and you see a whole bunch of thin bands. Okay. One of the things about the thick and heavy, thick bands do not mean that they are heavy fragments. Okay. Thick bands just means that there are many fragments of that size. Okay. So the heaviness of a fragment depends on its location away from the wells. If it's closer to the wells, like all these ones here, okay, that are at the bottom, if they're close to the wells, they are heavy. Okay. But if you look at the ones that are further away, okay, that just means they're light. Okay, so if they're lighter, they can travel further away from the wells. But if we look at a band, this one at the bottom here, you can see that this band is quite thick, and the one above it is actually even a little bit thicker. And then you see some bands, like over here, where it's a very, very thin band in comparison. Well, the thick and thin bands just tell you the amount. So imagine it this way. You have a large fragment, okay, that's trying to move through the gel. It only makes it up to this point here. Okay, so it leaves a mark. You have more than one copy of that fragment, so it keeps traveling up, 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 and as long as it's the same size, it will also deposit down here. Okay, so if you have more and more deposits, it's going to be a thicker band. So that means there are many bands of that size. The thin bands, okay, you can see them both at the top. You can see some of the thinner bands. They also occur at the bottom too. That just means that there are fewer fragments of that size. So we don't have a lot of light fragments, okay, of this particular size because the bands are thin. So the two things that you have to pay attention to: size of the fragments, okay, heavy fragments at the bottom, light fragments at the top. The thickness of the band determines how many of those fragments are present in your sample. So once you have that DNA fingerprint prepared, you can now use it to match up your DNA to whatever's found at the crime scene, or you can use DNA fingerprints to figure out if you're more or less related to another person. Because if you are related to another person, you'll likely have matching fragments, and depending on the number of fragments that match, okay, or the number of bands that match, you could be very closely related to that person, okay, or not. So people who are not related to each other usually do not have DNA fingerprints that are similar. Okay, brothers and sisters, okay, they will have similar DNA fingerprints. Not exact, but close. So DNA fingerprinting is a precise science. It's 99.9% accurate, okay, because humans are the ones who create these DNA fingerprints. So there's always a percentage, a small percentage, that there could be some errors. But because they're so accurate, even the courts of Canada accept DNA evidence, and usually if you have DNA evidence, that pretty much says that a person or a suspect was present at the scene of a crime. Okay, but just because you place a person at the scene of the crime doesn't mean that person has committed a crime. That's still for the courts to decide and for the lawyers to argue over. But because DNA evidence has been around for a while, it's a relatively new technology. So it's been in Canadian courts for about 30 years, okay, about 31 years to be exact. The very, very first case of DNA evidence being used successfully was in 1987, where DNA evidence was used to put away an Ottawa rapist. Now, DNA evidence has also been used to prove people being innocent as well. Okay, one of the more famous cases is David Milgaard. He was convicted of rape in 1969. Okay, but with DNA evidence, about 28 years later, he was exonerated from those charges. Okay, they were able to prove with DNA evidence that the semen that was found on the victim's underwear was not his. And if it wasn't his, that means that he didn't commit the crime. So I believe he got a pretty large sum of money from the from the government of Canada because they had wrongfully convicted him of rape. Like, he spent about 28 years in prison. Okay, and if it weren't for DNA evidence, he would pretty much still be in prison. So this type of evidence is very important, okay, and it's quite useful. So to summarize, basically you need to find a DNA sample. Most DNA samples that you can find are blood samples, semen samples, skin samples, hair samples, saliva samples. Wherever that sample is taken, we use restriction enzymes to cut it into fragments. We use the PCR technique to make more copies of that fragment. And then by inserting those fragments into the wells of a gel electrophoresis, okay, we can turn it on by running some electricity so that it will sort those fragments by size and mass. Once that is sorted, we basically get a pattern of DNA that you can use to compare. And that's pretty much it for DNA fingerprinting. It sounds simple, but the process itself is a little bit more complicated. However, it has worked so far in our Canadian courts, and I'm sure DNA evidence will either only get better and more accurate, and hopefully you guys will have learned something about forensic DNA as well. So if you're interested in forensic science, forensic DNA, okay, this is the area that you should probably get into and learn a bit more about. Alright, that's all I have. See you guys soon.